We are starting with the phenotype searches, and as you will see through the exercises, VEuPathDB integrates different types of phenotypic data, including data from high throughput studies, whether carried out by entities such as individual labs or more coordinated projects. We also integrate data from other resources that curate this type of the phenotypic data. We also perform internal curation of the phenotypes, and we accept and integrate data from the community. Today, you will take a look at the data sets in different resources. For example, you will investigate the fitness data in ToxoDB, where Toxoplasma gondii parasites expressing Cas9 and transfected with single guide RNAs were used to infect human cell culture. And the fitness and the phenotype scores were determined as the frequency of a single guide RNase library corresponding to the relative abundance of the parasites carrying the targeted mutation. And so this is also described as a screenshot that you will see here on the right. We also have data sets for piggyback insertion mutagenesis, where you can mine data using scores of the relative growth fitness cost for mutating a gene in Plasmodium. When it comes to looking at the localization data, such as cellular localization data in TripDB, we have integrated the data set from TripTag database, which aims to determine where every trypanosome protein localizes within the cell. And although I have included also a data set for Giardia, and although this is not a typical phenotypic data set, but is located under the protein localization data, but it's still a really cool data set to look at. In Giardia, this deals with subcellular localization of selected trophozoid or cytoskeletal proteins, where live imaging of GFP-fused strains were carried out using microscope techniques. So when it comes to integrating data from other resources, we have a data set in TripDB where it was incorporated from the Sanger, where it was a targeted manual curation of the phenotypes. In FungiDB, we have a data set that contains curated data for host and pathogens that was integrated from Phibase. And we also carry out, as I have mentioned before, internal curation of publications. We have a curation team led by Beatrice, who's on this call participating in the workshop as well. So if you have more information, if you have more questions about it, please feel free to contact her. And the targeted curation is carried out within the selected reference genomes. For example, we can take a look at the high throughput knockout mutant analysis and then characterize phenotypes either using controlled vocabularies or ontologies. So when it comes to internal curation and the approaches that we use, the phenotypes in general can be curated as using controlled vocabularies, for example, where text is described in where the phenotypes are described with text structured in a certain format, or you can also use ontologies. And in VEuPathDB, we use the entity plus quality or E plus Q approach, which is also known as post composition approach. And this approach utilizes different ontologies that are used to represent entity, which is the phenotype that is being characterized. For example, we have already looked at some of the phenotypes using Go terms searches, and also integrates quality information, such as how the entity or the phenotype is being affected. And you can imagine that for this type of analysis, you can utilize different types of ontologies, including PATO, or the phenotype and trait ontologies. If you're working with trypanosomes or parasites, you may utilize ontology for parasite life cycle to describe the phenotypes that you have observed. Or if you're working with fungi, such as ascomycetes, you can use the ascomycete phenotype ontology or APO to describe the mutant phenotypes that you see, such as defects in filamentous growth or absence of conidia. Now, the reason why we use the post-composition approach in VEuPathDB, it's because it allows more flexibility when it comes to data exploration. More specifically, this simplifies things a little bit and gives a little bit more power to you as a user when it comes to automated methods such as machine learning or interoperability and cross-references ontologies between different databases, such as that you can mine various ontologies in different databases and also build complex strategies, including the phenotype data. So coming back to the types of phenotypic data that we integrate in VEuPathDB, I also wanted to mention the data sets that we integrate as a community curated data. So there are several projects that we have worked with and facilitated. And here are a few examples of these projects in FungiDB. For example, if some of you ever worked in Neurospora crassa, a while ago, there was a project where researchers from different labs targeted the targeted genes in Neurospora crassa to generate a knockout collection, and then worked together to characterize these knockouts. And I think it was about over 10,000 genes, and characterization was done at different temperatures, different growth media, and also various labs collected image data such as phenotypic characterization of fungi growing on the plates, as well as the type of their growth and characteristics of the hyphae extending, for example, at the tip of the growth. And this type of data is integrated as a controlled vocabulary data within the gene record pages, where you can navigate to the phenotype section and examine the data from this project. Now, phenotype data or any other data for that matter that you feel should be associated with the comment can be posted on gene record pages as user comments. And here's an example of the user comment for Fusarium graminearum, where a particular gene is noted to have a reduced virulence in Fusarium, which is a plant pathogen. And these type of user comments are really cool, because if you submit a user comment, it becomes available on the live site essentially immediately after you submit the comment. And to facilitate entrance of the comments where you have more than, let's say, a handful of genes, maybe you're working with hundreds of genes, we do have a bulk user comment submission form that you can easily download, fill out, and send it back to us, and we will do the work for you. Now, we have also developed a phenotype collection form. In this case, I'm showing examples from FungiDB, but the phenotype collection form can be adapted to essentially any VEuPathDB database. And if you working on the phenotypic data collection or thinking to start a project, let us know, and we can help you we can work with you to help you guide you through the collection of the phenotypic characterization, such as how to characterize phenotypes using controlled vocabularies, or if you want to even give a shot to using ontologies, we can generate examples of how to do this. And the phenotype collection form at the moment can be downloaded from the how to submit data section in VEuPathDB. And there's a little bit more information within the form that you can read about and ask us questions if you would like more information. Now, at this, I will switch over to, I will show you two more things. Right now, we'll switch gears a little bit and talk about the interface that you are going to see when looking at the phenotypic evidence. So here is shown a screenshot from PlasmoDB, and I am on the Identify Genes based on the Phenotypic Evidence page. And this page contains several data sets, and you will notice that there are different data sets in different databases. And there's also a legend at the top that represents the type of the searches available for the data sets integrated in VEuPathDB. And these type of searches are listed at the top. So for example, association to genomic segments is a search for genes that looks for any genes that are associated with genomic segments by EQLT. The Curated Phenotype Search, or CP, returns genes based on phenotype characteristics. And for the data for which we do not have ontology incorporation, but rather have phenotype qualifier data as a text. This search will present you with a filter parameter to quickly choose the desired phenotype characteristics. There are two searches, similarity and similarity of association, located in the middle of the page. And the similarity is the search for genes which have a similar profile for an experiment. And similarity of association is a search for gene by similarity. For example, LOD scores that refers to numerical result when estimating whether two genes or a gene and a disease can be linked, can be used to set up cutoff criteria. And you can explore some of these data sets within the VEuPathDB databases. And finally, there's one more search, which is a phenotype text, and this is a fairly new implementation. And the phenotype text search returns genes whose phenotypic description contains your text term. For example, you may want to search for canidia or reduced phenotype depending on the species that you're working with. Right, and finally, I'll show you a quick search of the phenotypic data in FungiDB. And so here's, I am on the phenotypic evidence search that lists the available dataset in fungi. So notice that the type of the searches available here is slightly different based on the datasets integrated. And if I wanted to set up a curated phenotype search, I'll just click on CP and navigate essentially to the metadata available for this data set. So if I wanted to look for disease manifestation, I could check the criteria that I want to look. But in this case, I will go ahead and say, you know what, I want to identify any genes that have annotation in the disease and click on Get Answer. And so when my search completes the run, the window will get refreshed, and note that you have now about 2,000 genes corresponding to that parameter. So one thing that you could do, notice that right now you essentially have various species and various genes listed here. But if you wanted to look at the one organism, for example, Aspergillus fumigatus, a fungal organism that causes aspergillosis and also significantly reduces survival rate in COVID patients, you can click to recharacterize and find the genes in the AF293 strain, which is commonly annotated for these type of phenotypes. And if you want to expand the view of the results table, just click to hide the organism filter. And here you can explore results, download the results, and also add columns. For example, if you're interested in looking whether some of these genes that cause disease phenotype are also have non-synonymous SNPs, you may want to choose to activate the non-synonymous SNPs in all strains and update the columns. And the column will tell you whether there are any non-synonymous SNPs identified within the particular gene from the whole genome sequencing analysis. All right, so that's in a nutshell. That's it for the introduction to phenotypes. And I believe that if there's no other things, Amar, that you would like to mention, we are ready for breakout rooms.
Great. Yeah, I think we should just go straight into breakout rooms. There was a question in chat, but I think that got answered by Suzanne. So Rachel, maybe we can go ahead and open the breakout rooms, and so we can get started with the exercise.
All right.